Welcome to attending today's webinar. I'm Bradley Jiang, a study director from GemPharmatech, and the topic of today's webinar is drug resistance, tumor models in mice, mechanism, establishment, and applications. We're going to share to construct the preclinical drugs resistance models and the use of these models to evaluate the method for overcoming the drug resistance. And today's webinar contains 4 parts: the brief introduction of the drug resistance, preclinical evaluation of ADC resistance, preclinical evaluation of anti-PD-1 resistance, and the resources from GemPharmatech. First, please allow me to give a brief introduction of the drug resistance. Since oncology treatment has improved drastically over time, emerging treatment, targeted therapy, immunotherapy, together with surgery, radiotherapy, and chemotherapy, contributes to the 5 pillars of the cancer treatment. Precise medicine and early screening in conjunction with these emerging therapeutics have just dramatically reduced the mortality rates for the oncology patients. However, due to the complex biological characteristics of the tumors, different therapeutics approach ultimately move forward, making the tumor resistant to these treatments. From the source of the resistance causes, it can be divided into intrinsic causes, tumor cell itself, and extrinsic parts, including the basement membrane, vascular system, and immune cells. On one hand, due to the tumor heterogeneity and genomics instability, drug resistance closely eventually develop into major clone before or after treatment. And on the other hand, the tumor microenvironment is changing, especially the immune environment, allowing the tumors to become more drug resistant. And resistance is categorized as a primary resistance and acquired resistance based on the response to the drug before or after treatment. And the mechanism of the resistance is to different drug types are complex, including the epigenetic alteration, efflux pumps, genetic mutations, tumor heterogeneity, immune escape, and environmental support. In summary, the changing the physical parameters, the genome, and surrounding environment of the tumors drive drug resistance. This variety of mechanisms is the reason why it is too important to have the right models of the drug resistance. There are commonly following ways to establish the drug resistance models in preclinical models. The first way is to establish the PDX from the drug-resistant patient. However, due to the difficulty of obtaining and the scarcity of the resources, it's more convenient to use the mice to establish the drug-resistant models. We can obtain the drug-resistant cell lines by in vitro induction, and gene editing to introduce the drug resistance mutations. By these ways, we have successfully established a variety of drug resistance models, especially for the ADC resistance models and anti-PD-1 resistance models. Next, I will share how we establish the resistance models of ADCs. As we know that Enhertu has been approved by the FDA rely on this bystander effect, Enhertu has better inhibitory effect on both HER2 positive and HER2 low expressing tumors. Data from the clinical trials show that Enhertu dramatically improved the PFS of the patients, but still 24% of the patients still inhibit disease progress after 12 months of the treatment with Enhertu. To mimic the introduced Enhertu resistance, we inoculate the GMT1 breast cancer cell line into the mice and treated the mice with Enhertu. After the treatment, tumors that did not respond were collected, expanded, and reinoculated into mice following the Enhertu treatment, and these procedures were repeated for 3 times. Finally, the GMT1 Enhertu resistant cell lines was established, and we inoculate the nude mice with GMT1 parental or resistant cell line, and then treat it with Enhertu with 3 mpk once, and the results show that the GMT1 Enhertu resistant cell line significantly reduced the sensitivity to Enhertu. And meanwhile, the in vitro cell killing test also showed that the resistant cell line showed almost no response to Enhertu treatment. The resistance mechanism of Enhertu mainly consists 4 parts: antibody-mediated resistance, impaired drug trafficking, disrupt lysosome function, and payload-related resistance. Especially for the HER2-positive breast cancer, mechanism can be summarized as antibody-related and payload-related, and we explore the specific resistance mechanism of this model. For the antibody side, and the FACS analysis for the cell line and the tumor show that no statistically differences of HER2 expression observed between the HER2 sensitive parental cell line or the HER2 resistant cell lines. And this data also validated by the IHC staining of the HER2 expression and HER2 resistant GMT1 tumors standing positive for the HER2. And this result indicated that the resistance is not caused by the reduction of the HER2 expression. For the payload side, we also tested the sensitivity of the 2 different payloads, DXd MMAE, on the parental and the derived HER2 resistant cell lines in vitro. With the cell killing assay, the results show that the resistant cell line reduced their sensitivity to DXd and MMAE, which indicated that main resistant mechanism of this model may relate to the sensitivity changes to the payloads. To further clarify the molecular mechanism of this drug resistance, we conduct RNA sequencing and DNA sequencing within HER2 resistant cell line and parental cell line. Since the resistant cell line is isolated from the tumor trunk in mice with pooled clone, sequencing data can give us a hint to pick up the mechanism in this cell line. The RNA sequencing shows that some signaling pathways are upregulated in the resistant cell line, such as the transmembrane signaling receptor activity, external encapsulating structures, and the ABCG2, also known as breast cancer resistant protein, was upregulated in the GMT1 in HER2 resistant cell lines. And in DNA sequencing data, we found some relevant gene carrying the mutations such as MUC4, TOMP1MT, TP53BP1, and SLX4IP, which is SLC4 interacting protein. It has been reported that this silencing of the SL4 genes lead to the DXd resistance. This silence mutation of SLX4IP may also contribute to the resistance mechanism in HER2 resistant cell line. And to overcome the ADC resistance, the conventional methods include the combining with other drugs, increasing antibody recognition of targets by specific antibodies, and conjunct with different payloads. When we test another ADC on the cell line by replacing the conjugate of the HER2 with another payload and maintain the antibody part, and the in vitro killing test show that this cell line exhibits the increased test article sensitivity. Moreover, in vivo test also found that This new ADCs could significantly inhibit the tumor growth in GMT1 in HER2 resistant cell line. Next part, I'd like to share the experience in establishing the PD-1 resistance model. As the PD-1 treatment is more widely used in the clinic, our understanding of the mechanism of the PD-1 resistance is becoming more comprehensive. The mechanism was divided into two parts, the primary resistance and acquired resistance. Understanding the specific resistance mechanisms can help us make more specific resistance models. To be able to better mimic the human anti-PD-1 resistance in mice, we have three different kinds of PD-1 resistance models: the drug-induced resistance cells, gene-engineered resistance cells, and primary resistance cells. In drug-induced resistance cells, since the Keytruda is the immune cell dependent, we can only get the induced resistance models by in vivo induction. This method can provide a more accurate representation for clinical patient outcomes following the Keytruda treatment. We established the Keytruda-induced acquired resistance models in CD26 cells. CD26 cells were inoculated into PD-1 humanized mouse and then treated with Keytruda. Tumors showed no response or less response to Keytruda were isolated and digested into single cells, then re-inoculated in mice with Keytruda treatment induction. Re-implantation were repeated like 4 times. After that, we got this CT26 PD-1 resistant cell line. And here's the validation data. In CD26 wild type inoculated into PD-1 humanized mice, we found that Keytruda exhibits the very significant anti-tumor efficacy with a TGI of 76.8%, whereas in CD26 PD-1 resistant cells inoculated into the PD-1 humanized mice, the tumor showed no response to Keytruda treatment. We also tested the TILs after treatment to better understand the characters of the cell lines. In the induced resistance models, a percentage of CD4 T cells, CD8 T cells, and the macrophages were markedly reduced compared to the parental counterpart. The tumor infiltration of total CD45, CD4 T cells, CD8 T cells, Treg cells, NK cells, and the MHC2 positive cells were also decreased upon the Keytruda treatment. In addition, we observed a significant accumulation of GMDSCs in the resistant model after Keytruda treatment. And RNA sequencing data show that no significant changes were found in the expression of genes encoding immune checkpoints such as PD-1, LAG3, or CTLA4, but the upregulation of certain genes in the CD26 PD-1 resistant models, such as CCL2, CCL5, and CCL7, which encode chemokines, knows to promote an immune suppressive environment or contribute to the resistance to the PD-1 therapy. The second way to get the resistant cell line by using the gene engineered to modify the cell line carrying specific mutation, which lead to the resistance to Keytruda treatment in the clear manner. STK deficient has been linked to resistance to immunotherapy, particularly in patients with KRAS-driven lung adenocarcinoma. In the engineered PD-1 resistant CD26 cell line, we now call the STK11 in CD26, and this cell line showed no response to Keytruda. And STK11 deficient result in lower infiltration of CD8 T cells, along with significant accumulation of GMDSCs. Furthermore, this STK11 knockout was also associated with the PD-L1 low expression. Another engineered acquired resistance model, B2M knockout. But we know that B2M is a subunit of the MHC structure. And knocking out this B2M can lead to the impaired expression of MHC, leading tumor resistant to the anti-PD-1 treatment. We established these resistance models by knockout the B2M in CD26 cells. This model showed no response after Keytruda treatment, and the immune cell infiltration showed no significant changes, except the MHC1 expression is abolished. The third way to establish the model of anti-PD-1 resistance is the primary resistant cell. This model can better represent a patient who initially shows tolerance to the drug resistance. This primary resistance models contains many different cells on different background of mice, including the pancreatic cells, KPC, PANC02, myeloma, cell line B16F10, lung cancer, cell line LC1, breast cancer, cell line 41 and EMT6, kidney cancers, cell lines like RINCA, and some lymphoma cell line MPC11 and MPOC315. And the immune cell infiltration and the immune checkpoint expression should vary in these primary resistant cell lines, which indicates that every single cell lines has its own mechanism to resistant to anti-PD-1 treatment. For overcoming the PD-1 resistance, a common approach is combination therapy, which targets to overcome the underlying resistance mechanism through different combination therapy features. Next, I will share two cases study using these resistance models for overcoming PD-1 resistant therapeutics. The KPC cell line was obtained from a tumor mass isolated from a spontaneous pancreatic tumor mouse, KPC, and this cell line carry mutation in KPC mice, KRAS G12D, TP53R172H. And we tested the mouse PD-1 antibody on this KPC model and found that KPC tumor primarily resistant to this anti-PD-1 treatment. While showing the sensitivity to another antibody, CD40 agonist. This CD40 antibody was given in only one dose, as the mice showed a significant decrease in body weight after administration, but the KPC still shows very sensitive to this CD40 agonist treatment. And we can use this model to evaluate the co-stimulator factors overcoming the PD-1 resistance. In the STK11 knockout model, it has been reported that TNG216 rephrase the tumor immune microenvironment, reduce immune suppressive neutrophil infiltrations, promotes effector T cell recruitment, and reverse anti-PD-1 resistance caused by the STK deficient by inhibiting the activity of the KRAS HDAC complex. And we administered anti-PD-1, anti-mouse PD-1, and TNG216 alone, and in their combinations, in this BABC mice inoculated with CD26 STK11 knockout cell, and the results show that there was no response to the monotherapy alone, but the combination treatment exhibits the significant anti-tumor efficacy. Finally, to summarize the characteristics of different methods to construct models and the directive of the applications. For primary resistance and induced resistance model, these models, their resistance mechanisms are complex due to the tumor heterogeneity. Clones with different resistance mechanisms may mix in the tumor, and these models can better represent the clinical features. For gene editing resistance models have a clear resistance mechanism, it's good to validate the certain treatment with defined mechanisms. And we can choose the appropriate models according to the specific mechanism to reverse the drug resistance. At the end of the webinar, I will give a brief introduction of GemPharmatech. GemPharmatech is a high-tech enterprise. It specializes in generating genetically engineered mouse models and providing the trusted preclinical services to our customers. We are a global company with our headquarters in Nanjing, China. We have like 6 branches in China and a subsidiary in San Diego, USA. This can help us to respond more quickly to the need of the global market. One of the resources library is QAP project. Our knockout all project has constructed like more than 20,000 knockout and conditional knockout mice, as well as the 2 mice library. You can find your matching mouse model on our website. To better simulate human biological characteristics in preclinical animal models, we have established 5 humanization pipelines. These include the genetic humanized, immune system humanized, liver humanization, microbiome humanization, and antibody humanization. By relying on these humanized models, we can reflect the pharmacological response more closely to human characteristics during the preclinical drug evaluation. Relying on strong model construction capabilities, we also provide CRO services using these construct mice for one-stop solutions. Our CRO services pipelines covers oncology, autoimmune, metabolism, and neurological. We offer over 1,000 efficacy services and non-GLP safety assessment services per year, including over 200 projects for R&D applications. Our services including in vitro and in vivo efficacy, behavior testing, imaging analysis, and PK/PD services. Finally, I would like to share our clinical oncology services pipeline and resources. The drug evaluation covers a variety immuno-oncology-related treatment, as well as the conventional chemical drugs or emerging ADC drug evaluations. And this is all for today's presentation. Thank you.